In general, we'll look at DNA replication. We'll look at how DNA is replicated within the cell, where it's replicated, and when it's replicated. The enzymes we'll talk about during this session are DNA polymerase, ligases, helicases, topoisomerases, and telomerase. We'll also look at the utility of DNA repair, several different mechanisms of DNA repair, and why it's essential to maintain a pristine copy of the genome. Finally, we'll talk about DNA rearrangement. Different types of DNA rearrangement are both advantageous and disadvantageous within the genome. So let's revisit our central dogma. Here we have DNA, which in this case can be replicated. DNA can also be transcribed to RNA and be translated to protein. For this, we'll focus on DNA replication. We're going to look both at DNA replication in prokaryotic cells or bacteria, as well as in eukaryotic cells or mammalian cells. There are some similarities and differences to note. In both cases, it's semi-conservative. That's shown here, where in both cases synthesis involves a parental strand and synthesis of a new strand, such that one strand or one parental strand of the genome is always conserved. Synthesis in both cases requires primers. The primer in both cases is an RNA primer, and in prokaryotes, that is provided by an enzyme called a primase. In eukaryotes, the primer is provided by DNA polymerase alpha, and within that polymerase alpha subunit, there is a subunit termed the primase. For processivity, in prokaryotes, there's a single processing enzyme referred to as DNA polymerase 3. In eukaryotes, there are two different polymerases that are involved primarily in synthesis. DNA polymerase epsilon, which synthesizes DNA on the leading strand, and DNA polymerase delta, which synthesizes on the lagging strand. Following synthesis, the primers must be removed from the newly synthesized double-stranded DNAs. In the case of prokaryotes, the primer is removed by polymerase 1, and in the case of eukaryotes, the primer is removed by a family of enzymes, specifically RNase H, that hydrolyzes the RNA from the DNA strand. Just as a reminder, in prokaryotic cells, there is no membrane-bound organelles. Consequently, the DNA floats free within the cytoplasm, and this is in contrast to the highly structured chromosomal DNA within eukaryotic cells contained within the cell nucleus. The structure of DNA is also different between the two organisms. In prokaryotic DNA, the DNA is circular, similar to a plasmid, whereas in eukaryotes, the DNA is linear. The linear nature of DNA in eukaryotic cells generates a problem at the end of chromosomes. This problem is solved by an enzyme, telomerase, and we'll talk about that in a few slides. So the process of DNA replication between the two organisms is summarized here. Here, we can see eukaryotic DNA replication. There can be multiple origins of replication, denoted here, and multiple replication forks moving out in either direction to generate these newly synthesized strands in a semi-conservative manner. This is in contrast to prokaryotic replication, shown here, where the circular DNA has a single origin of replication. Again, the replication forks can move out in either direction until there are two newly synthesized circular DNA fragments. So when does DNA replication occur? What initiates DNA replication, and why would a cell need to replicate its DNA? Those are all very good questions. DNA replication is restricted to the S phase of the cell cycle, shown here. So when a cell receives a mitogenic signal, or a signal initiating it to proliferate or divide, the cell will undergo the cell cycle. The cell cycle is summarized very briefly here and involves this first series where a stimulus initiates the cell cycle. So a cell that is in G0 or is resting can be stimulated to initiate the cell cycle, and it will enter G1. During G1 phase, the cell will grow and initiate metabolism and other reactions preparing for the rest of the cell cycle. In the S phase, this is when a cell will replicate all of its chromosomal DNA. This is in preparation for G2, where the cell begins to align the homologous chromosomes and prepare for mitosis or cell division, shown here. So that's an overview of the process of DNA replication from a cell point of view. So what do you need for DNA replication besides DNA? As I mentioned in the general aspects, there are several proteins involved that are required for DNA replication, and this is true for both pro and eukaryotic organisms. You need a DNA polymerase. DNA polymerases are responsible for adding nucleotides to the growing strand, and this is in the 5' to 3' direction. DNA synthesis is restricted to the 5' to 3' direction. A primase, which again synthesizes an RNA primer, helicase, which separates the parental strands or unwinds the double helix, single-stranded binding proteins, and these prevent the single strands of DNA from reannealing before the new strand of DNA can be synthesized, topoisomerases, and these topoisomerases relieve strain in the double helix as it's unwinding. There are some generalized enzymes that are responsible or able to remove primers. And DNA ligase, which joins the DNA fragments by forming a phosphodiester bond between two adjacent DNA strands. And this is particularly important on the lagging strand, which is synthesized in fragmented forms called Okazaki fragments. The other proteins we'll talk about are pCNAs, or proliferating cell nuclear antigens, and these enhance the processivity of DNA polymerases. So this is a nice schematic of all of the enzymes we've talked about and their location during replication in the replication fork. This is the replication fork here. You can see that the helicase is unwinding parental DNA strand. It's dissociating the bonds between the hydrogen bonds between the base pairs, shown here in the schematic. The topoisomerase is relieving the torsional strain of the helix. On the leading strand, shown here, which is being synthesized again in the 5' to 3' direction, you have an RNA primer, shown here, and polymerase epsilon, which is synthesizing the strand. In the lagging strand, it's a little different. The lagging strand must be synthesized in small iterative units termed Okazaki fragments. Because of this, it requires multiple priming events such that the polymerase activity is maintained in the 5' to 3' direction. So here you can see the primase, part of the DNA polymerase alpha subunit, priming the DNA and being followed by polymerase delta, which can then fill in the base pairs. DNA ligase is responsible for generating a phosphodiester bond using the 3' hydroxyl and the 5' free phosphate on the added nucleotide. It's this 3' hydroxyl that's required for this ligation event. Consequently, this drives the necessity to synthesize DNA from the 5' to the 3' direction. This new phosphodiester bond is shown here. Remember, this hydroxyl group is on the 5-carbon sugar of the nucleotide. Again, the phosphodiester backbone shown here. C2 of the 5-carbon sugar, forming this phosphodiester linkage. Here are our base pairs again: adenine, cytosine, guanine, and thymine. The base pairing is adenine base pairs with thymine, and guanine and cytosine base pair. G and C base pairs form 3 hydrogen bonds, whereas adenine and thymine only form 2 hydrogen bonds. Consequently, it requires more energy to break hydrogen bonding pairs of G and C nucleotides than T and A nucleotides. Again, if you start with double-stranded parental DNA, an origin of replication is initiated, and the replication fork can move in both directions. It requires an RNA primer such that you maintain processivity in the 5' to 3' direction. Eventually, you'll synthesize 2 new copies in a semi-conservative manner. And if you notice, there's this overhang on the 3' end of both strands. This is a problem in linear DNA and consequently requires a telomerase. So again, DNA polymerases can only synthesize DNA in the 5' to 3' direction. This is because of the requirement for the 3' hydroxyl to be used in the phosphodiester linkage. These two features pose a problem at the 3' end of a linear chromosome. Consequently, there's nowhere to place a new primer to fill in that gap. If this problem is not solved, the linear chromosome will become progressively shorter at the end of each DNA replicative round. Those single-stranded DNA fragments are unprotected and will be privy to degradation and damage. The cell solves this problem by adding DNA sequences to the ends of chromosomes called telomeres. And these are small repeated sequences, and there are hundreds to thousands of them at the end of each chromosome. Enzymes called telomerases are able to facilitate the replication at the end of a chromosomal strand. Telomerase contains both protein and RNA, consequently, it can function as its own RNA template. It's complementary to the DNA sequence of telomeres, and this allows for telomerase to bind to the 3' overhang. Let's look at that now. The telomerase protein is shown here. It's a protein with an embedded fragment of RNA that functions as an RNA template. This RNA template is complementary to the 3' overhang, or the telomere. This allows for base pairing and synthesis in the extended region. Consequently, once this is extended, it allows for DNA polymerase to fill the gap that was previously left.